Yeah, thank you. Thank you. Let's see. Well, I want to thank the organizers for inviting me, and I'm going to tell you about our work on designing protein structures, functions, and assemblies, and then tie it together at the end about how this might relate to therapeutics. So the basic process of computational protein design in its simplest form. Before I do get to that, I should just say that everything I'm going to tell you about today has the following workflow. Everything begins on the computer with a computer calculation of a sequence of which is optimal for a given a desired structure or function. Of course, since we designed the protein, we know the amino acid sequence. We know the genetic code, so we can translate it back to DNA. And what makes this all possible and more exciting all the time is that the cost of gene synthesis, as you know, keeps plummeting. So we can test more and more designs all the time, which is a good thing, because the harder the thing we try to do is, the lower our success rate. But basically, we can test lots of designs. Basically, in all cases, we end up making the protein, assaying it for structure and function. I put this slide in because sometimes after talks I get the question, but do you ever do any experiments? All right, so in its simplest form, as I was saying, the protein design calculation consists of taking a protein backbone, or coming up with a new backbone in this case, and then searching over the possible amino acid sequences for this backbone, for that sequence which has the lowest energy when it's in this backbone conformation. And energy here is calculated. It's exactly what you expect. It's the sum of the van der Waals interactions between the side chains, the hydrogen bonds, solvation effects are taken into account. And it turns out that relatively quickly, one can come up with a sequence which has the lowest energy of any possible sequence for this backbone. Now, this sequence, while it's the lowest energy sequence for this backbone, it is possible that the sequence has another still lower energy state, and we need to get at that before we actually go and test it experimentally. So fortunately, over the years, we've been working on protein structure prediction using, this is everything I'm telling you is part of the Rosetta program we've been developing over the years, but one of the things that we started off with was the ab initio structure prediction problem. And we can take advantage of this by now taking the sequence we've designed and predicting its structure. And as you know, that's a rather formidable computational challenge, but we've developed a distributed computing platform called Rosetta at Home, which you can all sign up for and help us out, where volunteers help us test these sequences we've designed. And so we can now take the sequence that's designed and search through all possible conformations, or a large number of possible conformations, to determine whether that desired target structure is really the structure that the sequence is predicted to fold up to. And typical results are shown here. In this case, we chose 5 idealized versions of protein folds with perfectly straight helices, perfectly straight strands, perfect turns, and we used the first movie to identify sequences which were very low in energy in these structures. It's a little more complicated than what I show you, because you have to let the backbone move too, because for any fixed backbone there's no guarantee that there will be a sequence which is really low in energy. And once we got those sequences out, we sent them out to Rosetta at Home participants as I described, and the results are shown here. Each participant sends back a result which is shown as a single black dot on these panels. You can see we have a lot of participants. On this axis is the distance from the desired target structure in RMSD, and in this axis is the energy as calculated by Rosetta. So you can see that there's, well, as you know, there's a lot of local minima on a protein energy landscape, and so some calculations get stuck out here at high energy and high RMSD, but all 5 of the sequences which we've designed here have the property that the lowest energy structures that are found are very close to the design models. Trajectories that start with the design models are shown in green here. And probably because of this, when we make the proteins, we find that in a significant fraction of these fold up into very stable proteins that don't undergo temperature denaturation. When we solve the structures, they're really almost exactly right at both the backbone and the side chain level. So we can design new structures really quite accurately using this type of approach. And We can do that not only for different types of folds, but within a given type of fold, like the ferredoxin fold, a common fold, we can design different variants on the theme. So we can really custom design proteins to have whatever shapes we want. And these are design models, and these are NMR structures. This is work by Nobu and Rie Koga, and the NMR structures were solved in Guy Montaglione's group at the NESG. So we can make these proteins small. If we start putting in disulfide bonds, we find that with 3 disulfide bonds, we can robustly produce very small proteins, which again are very temperature stable. And I think these could have some real advantages for the applications I'll talk about at the end. And going even smaller, when you start thinking about cyclic peptides, the conformational space is small enough that during the design process, one can consider not only the state you want to design for, but all the other states too, and come up with a sequence which is very low energy in the desired structure. This is work by Vikram Mulligan. And so he's starting off by just designing peptides to have unique structures. So these are his designs, and these are on NMR ensembles of these designed proteins. So in every case so far, he's been able to make these are all peptides of 7 to 9 amino acids, and the structures come out right. So we can really control the conformation of these small peptides. And his latest set, which I can't show you experimental data on yet because we don't have it, obviously there's no constraint to L amino acids, so we can mix L and D amino acids. And obviously now we're trying to put binding functionalities in these. And I think these could have some real advantages sort of at the trade-off between small molecules and things like antibody drugs, because we can get very well-defined shapes and stable structures, but with much, much smaller molecules that will be easy to make and perhaps orally available. Okay, so we've started a project a number of years ago called Foldit, where the whole world can get into the act of designing proteins. And I just wanted to show you some of the recent things that Foldit players have been doing, which I kind of think of as protein art. This is a freestyle puzzle, which you all have to play. You get 70 amino acids, extended chain, and you fold it up, and as you fold it up, it interacts with other copies of the thing you're folding up. And you have to get the lowest energy oligomer. So here's a trimer that someone's come up with. Here's a tetramer. Here's a pentamer. Some of these look like things that you may have seen before, like this one, although not a pentameric version of it. Other things you almost certainly will not have seen before. When you get non-scientists designing proteins, they do non-canonical things. Here's a beta sheet surrounding helical bundle. And so we're just testing these now. We're very interested in symmetric oligomers, which for reasons that will become clearer towards the end of my talk. All right, so that's designing new structures. But none of you are really interested in structures. You're interested in functions. So that's what I'm going to talk about the rest of my talk. So the first problem is designing binding. And we approach this problem kind of like a climber would approach a climbing wall. So you've got the surface of the target. And now the first step is to identify handholds and footholds on the surface where you can place not hands and feet, but disembodied amino acid side chains, which are these stick-like figures. And then once you figure out where you're going to put your hands and feet, you got to figure out where you're going to put your body. So that means we either build a scaffold using the methods that I've described already, or identify an already existing scaffold that can hold all these things in place. The first problem we tried this out on several years ago was the influenza virus hemagglutinin. This is a blow-up of the hemagglutinin surface in yellow, and this is in the stem region. There's this conserved hydrophobic groove, and we designed 2 proteins. Actually, we designed a whole lot of proteins, but only two of them bound the virus. I told you our success rate is far from 100%. So here are these hands and feet in the groove, and here's the scaffold that's holding them together. In both cases, small helical proteins. Ian Wilson's group solved the crystal structure of these, and here's the hemagglutinin, and here's the design model and the crystal structure superimposed. So basically these small proteins are binding to the virus exactly as designed, even coming down to the designs. So while our hit rate isn't that great, when we get it right for the right reason. So since this time, we've designed a number of proteins which bind in the stem region of the hemagglutinin. We've also designed proteins that bind in the head region, the sialic acid binding site. And we've been able to trimerize these, so they essentially don't come off. Eva Strauch, who developed these, calls them flu glue. They're essentially irreversible inhibitors. These proteins bind a variety of flu strains. We now have a version of this one that binds group 2 viruses. And so using these, we have the first obvious application is diagnostics, because you can grab onto the hemagglutinin on one end and then read out with the other. And again, since we have a number of variants, we can get pretty high specificity for different strains. Working with Paul Yeager's group at the UW develop paper-based diagnostics based on these, which are looking pretty exciting. This is just showing detection of flu using these binders. And the other obvious application is therapeutics. So there's work from America Treats and Deb Fuller at the UW, who have basically been exposing mice to the California 09 virus. And if you give them the binding protein within 24, one of these binders, within 24 hours pre or post-infection, you see a dramatic decrease in weight loss and then morbidity. So it's early days with this, but it's looking pretty promising. This is with intranasal delivery. All right, so there's other viruses. Nothing I've told you about so far, and indeed nothing in the talk, is really specific to any particular target. So one of our big problems is, what should we be applying this technology to? Well, another obvious other viruses are the first obvious thing. So here's Ebola virus, and Erica Sapphire has done some beautiful work characterizing the binding sites of neutralizing antibodies on these. And here we've, in the site that's sort of analogous to this influenza stem region binder, we've designed a small protein that binds tightly to the this sort of fusion involved region of the virus. It's still in the early days. We don't have super high affinity, but we're pretty optimistic that this will likewise go. The previous binders were in the high picomolar range, and we expect these will as well. We're starting to think about the receptor binding site. Okay, well, it doesn't have to be pathogen proteins. It can be host proteins too. And so, for example, PD-1. Here's PD-1. We can use exactly the same procedure to design a protein which binds PD-1. We're still optimizing this, but we're at about 5 nanomolar now. It's a relatively small protein. And so if any of you have any interest in PD-1 binding proteins, definitely let me know. Now, what about sort of biotechnological applications? For example, purifying of antibodies. And so we've designed a protein which binds to, this is the FC region of IgG, and there's a histine here. And we've designed a protein that, Eva Strauch designed a protein which binds to this FC region and covers the histine. And so basically the result is that at low pH, this is, at high pH, when the histine is not protonated, there's quite high affinity binding. But when you drop the pH to 5.5, the binding is lost. It becomes it's above micromolar, or it's micromolar or worse. So if you look here, if you compare protein A binding to IgG at pH 5.5 and pH 8, the binding affinity is pretty much the same. Again, it's in the same ballpark as our design protein, but at pH 5.5, this protein doesn't bind FC. And this is just another picture of this is how protein A binds, this is how our design protein binds, covering up this histine on FC. And so if you make a column where you, and you load through a mixture of IgG and other stuff, on a protein A column it sticks, and then you wash it pH 5, nothing comes off. At pH 3 it comes off. With this protein, when it binds, when you elute at pH 5, it comes off. And it's very stable. You can purify it by boiling bacteria expressing the protein. So if you have any interest in purifying antibodies, let us know. All right, so what about intracellular interactions? You could say, well, come on, going after these extracellular binders, what's the point? Because you can get them with antibodies. But their intracellular interactions are a little harder with antibodies. And so I'll give you 2 examples of how we can make proteins that are very, very specific. The first one is, as you know, MDM4 and MDM2 both target P53 for degradation, but it's been hard to separately tease apart the roles because all the small molecules block both of them. And the second example is with BCL2 family containing members. And in both cases, what we're trying to do is design proteins that are very selectively bind to one family member, but not any other of the family members. So here is Here is MDM2 and MDM4 superimposed. You can see they're very, very similar. That's why the small molecules don't distinguish. James Moody in the lab has designed a protein, this blue one, that kind of hugs the surface. And he's got variants now that are very, very, because of all this extra, all these extra interactions, he can tune it. This variant is quite selective for MDM4 over MDM2. And he likewise has variants that are selective for MDM2 versus MDM4. So in collaboration with Mike Dyer and Brenda Schulman and others at St. Jude, well, they've basically done these experiments. They express these proteins, and just look at the first two lanes here. This is the MDM2 specific binder. It pulls down MDM2 in this band, but the MDM4 specific binder doesn't pull anything down. Whereas the MDM4 specific binder doesn't pull down MDM2 from cells, but does pull down MDM4, often referred to as MDMX. So you can start teasing apart then the separate roles of MDM2 and MDM4. Here's the BCL2 family example. BIM is a peptide which binds to many different BCL2 containing proteins. Here what I'm showing is the BHRF protein, which is made by Epstein-Barr virus. And Eric Prakos designed a protein which binds very selectively to this BHRF1 protein, but not to other BCL2 family members. And so he calls this protein BINDI. First of all, you can see that the BIM peptide binds to all of these different family members, in the viral one and the human host ones, whereas BINDI binds very tightly to the viral protein, but much more weakly to the host proteins. Eric's now going on to make binders, specific binders, for example, for MCL1 and other members of these families. So if he adds these AW cells are transformed with the virus. If he adds this, sorry, let's see, I'm getting this backwards. Let me just skip over here. So here we have control cells, and here we have control cells transformed with the virus. If he adds this design protein, which is BHRF1 specific, it induces apoptosis in the transformed cells, but not the untransformed cells, which is what you'd expect because it's specific for BHRF1. And this side just shows the same thing with cytochrome C release from isolated mitochondria. Now, the big problem, of course, with designing proteins to go after intracellular targets is delivery. And that's something I'll come back to. This work's done in collaboration with Pat Staten's group, who's done some very elegant work with polymer-based delivery. That's how these proteins are getting into cells. All right, so that's protein binding. The next topic I'll talk about is small molecule binding. So here we approach the problem in the same way. You take a small molecule and sort of come up with what you think would be an ideal binding site, making hydrogen bonds to the hydrogen bond donors and acceptors, and then building a complementary pocket. And here's the design of such a binding site here. Here's side chains coming out, making specific interactions with this ligand here. This is showing how shape complementary the design is to the ligand. And when we make these proteins, again, we have to test a number of them, but we do get binders. This is a crystal structure of one of them. The crystal structure is in cyan, and the model is in a magenta. And you can see that this protein is binding the small molecule for the right reason. The structure isn't quite identical to the ligand, but it's very, very close. We're making all the programmed in interactions with the ligand. And I won't go through this in detail, but since we've designed the site, we can control the specificity of the designed protein for different small molecule targets by incorporating or deleting hydrogen bonds, as shown here. So here's a series of related steroids, and we can reprogram the specificity of this protein for different steroids just by changing these groups. So we're now using this approach to generate binders to a variety of small molecules. We're interested in proteins in small molecules for two applications. The first is therapeutic sponges. If you get an overdose of a small molecule like digoxygenin is used to treat heart disease, if you get too much, it's bad. So there's, for example, a new family of blood thinners which have that same property where there's a relatively narrow therapeutic window. So we're designing binders for those type of molecules, as well as for molecules which there's general interest in sensing, such as cortisol. And we have some exciting results with collaborators with incorporating these proteins into both in vitro and in vivo diagnostic devices. I just wanted to say that protein design isn't restricted to using the natural 20 amino acids. I showed in one of my first examples using mixing D and L amino acids with peptides. With proteins, it's a little harder, but with Pete Schultz's technology, we can put unnatural amino acids like bipyridine into proteins and now design sites based around these. And that is such a site is shown here with a bipyridine chelating a metal with two other residues forming part of the design site. Now, an obvious issue with all these designed proteins, if you start saying design proteins and therapeutics in the same sentence, is immunogenicity. And what we're doing to try and get at this is to incorporate into the Rosetta design process, where I described before, we're searching for the lowest energy sequence for a desired structure or function. We can incorporate basically an MHC peptide binding predictor into the process, and also reward matching of human 9-mer sequences. We have a couple examples now where this has worked pretty nicely. The first is with GFP. This is the superfolder GFP. It has a rather immunogenic peptide here. We can now go through, basically redesign this part of the protein, trying to eliminate that peptide and other predicted MHC epitopes. And then when we make the protein, it's put in. We can see whether it induces a T-cell response. It activates T-cells. The original native protein gave a strong response, which is shown here. The deimmunized protein, that epitope is gone. A more practical example has been a collaboration with Ira Paston's group, who's been working on exotoxin A for a number of years and been trying to make it immunologically silent. They've made a lot of progress on this, but there were a couple epitopes that they weren't able to deal with. And we use the same approach to redesign these regions, preserving function, which is binding to its target shown here, but trying to eliminate these MHC interacting peptides. And this just shows results for a couple of these mutants, which retain their cytotoxic activity, but lose their T-cell reactivity. So we're pretty excited now about this method, and we're interested in applying it to other types of proteins. So if you have proteins that are giving immunogenic responses and want to try this out, please let me know. Okay, so the final thing I'm going to talk about is self-assembly. And here the idea is, can we make, I've talked about designing monomeric proteins, can we design assemblies of proteins? And so here is, this was our first result. This is work by Neil King in the lab. He designed a protein subunit that self-assembles, on the computer-ready way, into this cube-like structure, an octahedral cube. And in the electron microscope, what you see is a completely homogeneous field of particles. These are about 14 or 15 nanometers in diameter, and this just shows cross-sections, or just averages of these images. They look like little dice. We're trying to get to the point where we can put different numbers on different sides, but we're not quite there yet. When the crystal structure was solved, it was found to be virtually identical to the design model, with an RMSD of 0.7 over the full structure. And I think we've seen this with more results, as I'll show you, and we think it's because of the symmetry. It really really pushes down the error in the design process. So this is that same crystal structure. This is the design model. the crystal structure down 3 different axes. Here's another one, a tetrahedral one that Neil and Will made. So these are made of a single component, and they self-assemble in E. coli. So you make the protein, and it's just, when you open up the E. coli, you just have these particles. But suppose you wanted to package stuff inside, like small molecules, for example. It would be much more convenient if they were made out of 2 components, which you could then mix together with a small molecule and in so way package. So Neil and Jacob and Will went on to design 2 component versions, and I'll just take you through this one a little bit more slowly. Here's 2 inverted tetrahedra, and there's a green subunit which is at the vertex of each of these this tetrahedron, and a blue one that's at the vertex of this one. And now when you slide them together, you have a degree of freedom sort of in and out, and then you can also twist each one around its axis. So we do this with many, many choices for the 2 trimers here, the blue one and the red and the green one. And we search for arrangements which are very shape complementary. And then it's a design problem, pretty much like what we saw in that first movie, where we redesign that interface to get high affinity interactions. And then we can express the proteins either together or separately. So I'll just zoom through this. I hope you can see these pictures. This is for 5 different designs, which are shown here. We get these, here's just showing the field of view of the nickel purified protein. And you can see, again, you get these absolutely homogeneous particles. They look like little bite-sized shredded wheat in this case. And I'm not sure how many of these others you can see. Crystal structures have now been solved in Todd Yates's group by Dan McNamara for 4 of these, and they're shown here. Here's the design down 2 different axes, and the crystal structure shown here. You can see in all cases these design models are coming out. These designs are coming out very, very, very close to the model. If we make the components separately, they're stable, we can then mix them together, and they form these particles, and the particles that are formed are identical to the particles that are formed without them. We've been able to package proteins like GFP inside in collaboration with Don Hilvert, and the particles don't change. So applications for these, we're now making icosahedral versions of these, and we're very excited about these for targeted delivery. For one thing, these are totally homogeneous. Every particle is completely the same, and they're biodegradable. They're just made out of protein. I've shown you earlier in my talk how we can make these little binding domains that bind tightly to targets. Since we have 2 components, you could even imagine doing coincidence detection where you have one component on one and the other component on the other. And of course, what you would put inside could be a small molecule if you're targeting tumors. It could be protein. It could be RNA. And we're starting to play with these options now. Another application which Bill's going to give a beautiful talk on in a minute is to vaccine design. And so you could imagine with the 2-component cage, you could put, say, an antigen on one and an immune stimulatory component on the other, and maybe package an adjuvant inside. And again, stay tuned for what Bill's going to tell you beautiful stories about vaccine design. So that's what we can do with particles. What about, there's other types of symmetries, though, open symmetries, like 2-dimensional layers. And we can use exactly the same approach. And this shows, essentially coming straight out of E. coli, what happens if you make a protein subunit that's been designed to form a 2-dimensional sheet. And you can see it's forming this honeycomb lattice. We don't have a high-resolution structure yet, but if you make Fourier transform this and then generate a map, it fits this density almost exactly. So I think we'll be able to design layers which you can manage as protective coatings, all kinds of things, maybe encasing transplanted tissue. All right, so that's all I have to say. I've been really fortunate to have actually spectacular co-workers. So Nobu and Rie Koga did the work on the new scaffolds, Vikram on the cyclic peptides, Neil, Jacob, and Will on the self-assembling particles, Shane and Tamir involved in the work on the 2-dimensional layers. Protein binding, the flu work was done by Cyril Fleischman and Tim Whitehead a number of years ago. It's been picked up by Aaron Jorgen more recently on the stem, and Eva's done work on the head region binders for flu glue, and she's also designed the FC binding protein. Eric did the work on the BCL2 family binders, Cassie on PD1, James on MDM2 versus MDM4 discrimination. Chris King is developing the Rosetta deimmunization protocol, and Dave Law is working on Ebola. Christy and Sagar and Jai Dao have done the work on small molecule binding. And we've had a whole host of really fantastic collaborators too. So I'll stop there.
Thanks very much, David. Any questions? Please come to the microphone. So when you hear a talk like that, you think, well, you might as well just go home and give up. But I think it's probably not that bad in the sense that how many of the designs, or what percentage of the designs actually work first time, and how often do you have to make libraries and then select using sort of phage or yeast display for functional molecules?
Oh, yeah, right. The computation is very, very far from perfect. So when our initial designs are generally fairly weak binders, that we get the binding mode right, as we find out when we get the crystal structure, but invariably there's small fine-tuning adjustments, often electrostatic interactions, sort of not exactly at the interface. So we actually, for all of our binding applications, we make proteins, we basically put the gene straight into yeast, use Dane Wittrup's yeast display method. Actually, Dane's been a great help with this. And so, we're basically, as soon as we get a binder, the first experiment we do is to make a saturation single site mutant library where we mutate every amino acid to every other, do a round of selection just by facts, and then deep sequence. So we can basically see where the design was right and where we missed something that could improve affinity a lot. So we can feed that back to improve our design process, but that's also how we can jump to much higher affinity quickly.
So do you think you're going to ever reach a situation where you won't have to do that? You'll be able to do it on the computer?
Yeah, I think so. I mean, we'll get there eventually. I don't know how fast. But in practice, that's a pretty fast process. So yeah, so I think we'll continue to learn, but then we'll probably continue to do harder things. So our success rate will go down. So yeah, we're clearly getting better. There's a lot of, proteins are soft and there's a lot of subtlety. So I don't think it's going to be immediate that we'll be able to dispense with experiment altogether for optimization.
Okay, thanks very much. Question there?
I don't know if this is on. A couple of questions, David. So I can't help asking, having been out of the field for a few years, what do you think have been the major changes, the most dramatic evolutions of the technology to enable this kind of accuracy?
Well, yeah, let's see. Since you were doing stuff, well, the force field has been getting better. It's a slow, incremental process. But I think the main thing that's probably changed is we've gotten a lot more feedback, right? So we see what we're constantly learning from the mistakes, and that's the great thing about design. You make stuff, and most of the time it doesn't work, so you're always learning. Computing power, there's a lot more computing, so we can do this kind of testing designs by forward protein structure prediction. Also, gene synthesis has gotten cheaper, so we can routinely, if we're trying to design a binder to some new target, we can, without thinking about it, order 20 designs. And things like yeast display, Dane's developed, have really enabled this. We can order genes on chips now and screen them all in parallel by fax. So, and then the design methods have probably gotten better. I mean, none of the methods for doing any of this stuff did we have, back when you were doing this. So it's probably sort of been methods development, it's been gene synthesis costs coming down, it's been parallel developments like yeast display. It's been a lot of things.
Okay, all right. And if I could ask, I can't help, for the Rosetta at home, are there particular individuals or particular groups that tend to always do very well on structure prediction? Yeah, well, you're probably referring to Foldit, because Rosetta at home, basically, people are just contributing spare time on their computers. And actually, Foldit came about because people started writing into the Rosetta at home message board saying they were watching the screensaver and they thought they could do better. So Foldit is kind of like an interactive version where you can show how much smarter than you are than the computer. And there are top Foldit players. It's really been fascinating because people form teams, and the teams compete with each other, and they form teams in different time zones around the world. So we give them a week for puzzles, so they hand off solutions. And so when we've gone back in and looked at really amazing things, you can sort of see this handoff of stuff.
Another funny thing has been publishing. We've broken some paradigms of scientific publishing, because now when we've asked Foldit players whether they want to be authors on the scientific papers that have come out of what they've discovered, they've usually said no, just reference our team. So now you can look up, Foldit Void Crushers team is like is indexed in PubMed.
So are scientists any good at this?
Well, I think if it's your day job, it's just not as fun. So most of the people doing it are not practicing, biochemists or molecular biology. It's something that you do if it's really different from your day job. Actually, there's a really fascinating video of interviews with Foldit players, and you can see exactly what chords it strikes, and people who, they have kind of a day job, and it's not so exciting, and then they go home and play this game and get involved with people all around the world who are going to, cure cancer or whatever. We don't say that, but.
Okay, no more questions. Thanks very much, David.